Good morning, everybody, and welcome to our May 2024 AnVIL demos. Today, we have Elizabeth Humphreys, who comes to us from the Fred Hutch Cancer Research Center, who is going to demonstrate the single-cell with bioconductor activity that is part of our AnVIL demos book, a collection of activities that showcase a couple of uses of AnVIL. After the demo, we'll have some time for Q&A to answer any questions. We've got a couple of upcoming demos slated with dates for the rest of the year. So stay tuned for those topics as they get scheduled. So without further ado, I'm going to turn it over to Elizabeth to talk about single-cell bioconductor in AnVIL.
Yeah, thanks, Natalie. Okay, I am going to share my screen. Perfect. And so can everyone see my AnVIL workspace open right now?
We see it.
Okay, awesome. Brilliant. Okay, so like Natalie mentioned, I'm going to be running through a single cell analysis option with AnVIL using the IC package from Bioconductor. And if you are interested in following along, you can find the instructions for what I'm doing in our AnVIL demos book. Specifically, it's the single cell with bioconductor activity down here. So if you go to hutchdatascience.org/AnVIL demos, you can get all of the instructions, including screenshots telling you exactly what you'll need to do. To make things a little more streamlined for this demo, I've actually already opened my workspace. It's the demos single cell bioconductor workspace, and I've cloned it, and you can see the name of the workspace right here. I've also already started my environment. We are going to be working with R today. And you can take a look and see that this is R Studio 4.4, Bioconductor version 3.19, and also it has Python version 3.10, although we're not going to be working with Python today. Okay. All right. So when you open up the workspace, down in the files, there is an IC R Markdown file that's associated with it. And if you click on that, it's going to bring up a Markdown file which has all of the code that is necessary to run this activity, as well as some information about the data we're going to use today. I did not create this data. This is actually from a bioconductor workshop in 2020. And it uses the PBMC 3K dataset from the 10x PBMC data package, or it's gene expression profiles for single peripheral blood mononuclear cells. I've already pre-processed the data based on the instructions from the workshop, and have saved the data as a summarized experiment object that is associated with this workspace. When you go through this activity, first you'll load your AnVIL library, and then you will bring the data, which is in an R data file. You'll associate it or with your persistent disk, or bring it onto your persistent disk, and then you load it into the workspace itself. I've already done that, and I've got a single cell experiment object with an awful lot of elements saved as the object SCE, or single cell experiment. I've already also installed the IC package. This doesn't come preloaded with AnVIL, but you can pretty easily and quickly install it using the Bioconductor install command. Okay. And I've opened the IC library. So the next thing we're going to do is actually just launch the IC viewer. When I do this, it actually opens up another screen entirely. And I'll make it larger so it shows up, it takes the whole screen. And when this browser or viewer opens up, you'll notice that it loads, I believe it is 10 different plots and tables. These are all the things that you can make with IC. And it's just showing you the options, and it is randomly filling them with the first row in your data. As you can see, it takes a little bit because my data file is very large. But here you go. So we've got, let's see, a PCA plot. We've got a table of the row data. We've got a plot of the features. In this case, the features are the genes that we're measuring the gene expression of. Can scroll down, and now we've got plots of the columns, plots of a row, plots by sample, a table of the columns, and then a heat map. None of these look particularly exciting right now, and that's because we haven't actually put the interesting data up there. So this is the default. And if you want to customize it, you can come up to this button up here. This is your display panel, and if we click it, it lets us either organize the panels or examine the panel chart. With examining the panel chart, it's going to just open up basically links telling you where or what sort of plots you have. That's not super exciting. But if you open up organize panels, now it's going to let us customize what we're seeing. And to start with, let's try making a Hinton plot. And so I'm going to delete basically all of these options except column data plot 1. Okay. And I'm going to actually change the width. So the widest it can be is 12. That'll take up the full screen, and that's what I want. So I'll choose that, and then I'll say apply settings. And you can see what will happen next is our viewer changes to show only the data plot that I've chosen. In this case, a column data plot. Now, I'm not done yet. I mentioned we're going to make a Hinton plot. And for those of you who aren't familiar with what that is, this is essentially going to be a plot that shows us which clusters, or how we can, the identity of the cell type by cluster. All right, so we'll go to data parameters, and we're going to first set our y-axis. And our y-axis, we are going to pick, scroll all the way down. We're going to put cluster. So on our y-axis, and then we're going to change our x-axis to column data. And we'll scroll down, and we're going to pick the labels. And you can pick either labels fine or labels aren't, but we'll do labels fine here. All right. And I can close that and scroll up. And now I can see I've actually got something rather interesting. On my y-axis, I've got clusters 1 through 14. On the x-axis, I've got the different types of cells that were found in this particular data file. And in the plot itself, I've got a dot that represents each cell that was in our sample. And if you remember, there were over 2,000 of these. So there's an awful lot of dots on this. And right now I'm hovering over basically our biggest plot. Yeah. This is a cluster of cells that have been labeled cluster 4. And we can see that most of the cells in cluster 4 are what are known as classical monocytes. Cluster 4 also has some other cells like intermediate monocytes. We've got, I guess, dendritic cells and naive B cells, just a couple of those. But primarily cluster 4 is our monocytes. And for anyone who isn't certain what or doesn't remember what a monocyte is, this is essentially these are white blood cells. These are the precursors of macrophages. So they are going to be attacking bacteria or foreign objects. Okay. All right. So like I said, this is a hidden plot, but maybe we want to take a closer look at the cells that make up our cluster. And we can do that if we go back up to our panel organization tab right here, and we go to organize panels. We can add something to our column plot or our hidden plot. By clicking here, let's take a look at, let's do a column data table. This is going to let us look at the columns or the data that we use to make this plot. And I'm going to again make that table width of 12 and hit apply settings. And the original plot I created is still there, but the viewer has now expanded. And as I scroll down, I see I've now added a data table. And for this data table, I have each row represents a different cell in my sample. And I can actually modify, there's several of these columns which we might not be interested in. You know, not all of this information is necessary. So if I go down to data parameters, this is going to let me customize my table. Oh, here we go. There we go. Specifically, it lets me hide columns that I don't need to see. So for example, I don't need to see barcode. I maybe don't need to see the sequence, and maybe I don't need to see the cell ranger version or the library. Each time I add a column to the hidden column option, it recreates that table. And when we go up there, we can see that all of the information I'd said I don't want is gone. And instead, I'm left with some of the other information I want. Now, if you remember, we were working with cluster 4. So if I want to see only those cells which have been assigned to cluster 4, I can actually sort by that. And it'll take a little bit of time because I am sorting through data. But now I only have 362 cells out of my more than 2,000. And these are all 362 that have been assigned to cluster 4. And we can go through and we see that label's fine. Yeah, I've got a lot of intermediate monocytes, classical monocytes, which is exactly what our Hinton plot, that's this plot up here, told us. Most of my cells are going to be classical monocytes. And then we'll also have some intermediate monocytes. And if I want to know exactly how many in cluster 4 are intermediate or classical monocytes, I can if I spell it properly, that's how you spell it. Oh, I've misspelled it. You can, however, if you have a little bit more time, you can play around with that and you can sort to look at how many cells you have that are just in cluster 4 and also classical monocytes. Okay, Well, that's all well and good. But perhaps you've been doing this experiment and you are curious about the gene expression of certain genes in your data set. We can actually create those plots too. So again, go up to your panel organization and go to Organize Panels. And I don't want my table anymore, so I can just remove my table. And maybe I don't want my Hinton plot anymore, so I can remove that. And instead, I'm going to look at a feature assay plot. The feature assay plot in this case is going to be gene expression levels of a gene. And it's going to be my only one, so again, I'll make it 12, the width of 12, and I'll hit Apply Settings. And now I've got a feature assay plot. This is not a particularly interesting one. This is for the gene that this is, what is that? MIR 1302. Doesn't seem to be expressed all that much in whatever my default x-axis is. But we can fix that. So if you scroll down to data parameters, click on that, it's going to let us choose whichever gene we want. And in this case, I'm going to actually pick a gene that is a marker gene for classical monocytes. It's called LYZ or LYS. It's short for lysosome. And I want my, I'm going to use log counts as my y-axis. And for my x-axis, I'm curious to see if indeed those cells, this gene is more highly expressed in my classical monocytes. So for my x-axis, I'm going to put cluster again. Remember, cluster 4 is the one that was supposed to be classical monocytes. So we close that up, and now we've got a violin plot looking at gene expression across all 14. I do have elevated expression in cluster 4. I also have elevated expression in cluster 9 and cluster 13. And that's interesting. If I've forgotten what those labels are, I have two options to see what sort of cells were in cluster 4 or what sort of cells were in cluster 9 or 13. I've got a couple of options. I can go down here and I can color all of my data. I can color it by column data and then choose down here either labels fine or labels on. And now I've got them colored. This isn't quite as helpful as I might have wanted. So instead, another option is just to recreate my Hinton plot. So I go down or go to my panel settings, and I again pick column data plot. And I'm going to have it change the width to 12. And this is actually going to put my Hinton plot or my column data plot right underneath my feature plot. All right. So there is the gene expression of the lyse gene. And down here, we just recreate that Hinton plot we already made. So our column of interest is again going to be cluster. And our x-axis is again going to be label fine. All right. So Cluster 4. And all right. Oh dear. Okay. So one thing you might run into that at some point something might happen and your oh no. Okay, that was a slightly embarrassing pause, but this actually, I think, is going to be very useful. I'm going to share my screen again, and I'm going to show you what happens if you are working on the cloud on or working on Anvil and your internet connection suddenly cuts out, like mine did. Okay, so even though my internet connection cut out, I still have my active workspace or my active environment, and nothing has disappeared. So I've still got my single cell experiment, or my data are still loaded. I actually still have all the libraries I loaded. So all I need to do is open IC again. All right, and it's going to launch another viewer. And while the plot I was working on did disappear, I can recreate that with no problem because my actual R session hasn't stopped. Yep, here we go. It is one of the very helpful things. I know when I first started working in the cloud, I was a little concerned what would happen if my internet got a little dodgy, and it turns out not very much. Uh, which is nice because if you're like me, you've had a terrible experience where all of your data disappeared at one point and you lost all that work. Okay. So we're gonna recreate that feature plot, and then I'm gonna show you how you can save what you've made. So go up to organize panels again, and I'm gonna delete everything except my feature plot. And in fact, I'm gonna make 2 feature plots because so I mentioned the lyse gene is used as a marker gene for classical monocytes. It's not the only one. There are actually a couple of genes that can be used as marker genes. And let's look at 2 of them side by side. So we're going to make the feature plot 1 the width of 6, and the feature plot 2 is another width of 6. And I'm going to apply my settings. All right. And on the left-hand side, I'm going to do the gene lyse. So we'll scroll down. And I'm going to change the y-axis to the gene LYZ. And my x-axis will be column data, and I'm going to change the column data. I want to look at it by cluster. Great. And we can see, again, we've got elevated gene expression in our clusters that we know have classical monocytes. So cluster 4, cluster 9, and then cluster 13. And another marker gene is actually CD14. So I'm going to change my y-axis feature on the right-hand plot to CD14. And I will change my x-axis to the column data. Or sorry, and that column data, I'm going to choose the cluster column. And now I'm looking at gene expression of two marker genes side by side. And you can see, as we were hoping, that the two clusters with lots of classical monocytes, both cluster 4 and cluster 9, have elevated expression of both of my marker genes. Awesome. All right, so you've gotten this far and you've made some plots that you'd really like. IC actually has the option, a couple of options for saving your work. If you go up to this button, this middle button up here that has the download symbol, we click on it, we can straight up just download these panels we made by doing download panel option. And that is going to, if I hit download, it will actually save it to your computer, not to your workspace. So that's pretty cool. But maybe I don't want to download the panel. I actually just want to know what R code was being used to create these plots. So I have the option of extracting the R code. And that's going to tell me exactly the code that I would need to run in the future to recreate what we've just made. And so it's here. It loads the libraries, the dependent libraries I need, and then it creates each plot without me having to type all of those options. I really like that. Frankly, I can never remember all of the options for ggplot, so I'm glad that this just puts it there for me. And if you happen to want to just know what sort of settings that you created, you can download just the panel settings as well. And it's going to tell you everything, all those options that you chose. All right. Yeah, so that is IC. And as you can see, haha, I didn't mean to make a pun. That was a terrible pun. Anyway, so this is how IC can work for you on AnVIL and can hopefully make some of your data visualization needs a little easier to handle if you happen to be working on the cloud. And if you close it off, you can just go on to your workflow, and when you're done, you can just delete your environment. And you can either save the persistent disk, so save the data you're working on, or delete the whole thing. All right, that is all I have for you today.
Thank you
Are
so
there any
much, Elizabeth.
questions? Yeah, of course.